Good afternoon, everyone. I am Jordi Esteve, hematologist at Hospital Clinic of Barcelona, but my duty today is to introduce, it's the privilege to introduce Dr. Mariona Graupera, a researcher that recently has incorporated to the Institute Josep Carreras. Mariona her research career is devoted mainly to the study of the vascular formation in healthy individuals, but especially in disease, in cancer and other diseases. She started her career at specifically at the hospital clinic, at the hemodynamic unit of the liver disease department with Professor Joan Carles García Pagán and Jaume Bosch. She performed her PhD there, and after that she moved to the Ludwig Institute of Cancer Research in London to perform a postdoc with Professor, I don't know if the pronunciation will be perfect, but something like Van Heijsbroek, which I think it's from a Dutch origin. And after that, she moved back to, and she established as an independent researcher at the EDBA. And she has stayed there for several years, and now she has moved to the IJC. I would like to highlight some of her main contributions to research, especially she has described PI3K signaling for vascular formation, which I think is a very interesting topic. There she has described several of the isoforms that are involved in this angiovascular formation, even in some tumoral disease as prostate cancer. I would like to know about her research in hematologic disorders. She has also studied, analyzed the crosstalk between tumor cells and the microenvironment, including the vascular formation, and well, many of other things that she will explain us in a few minutes. All this research has translated into several high impact factor publications in top research journals such as Nature Communications, Journal of Experimental Medicine, Cancer Research, Circulation, and many others. And also she has been awarded with several competitive grants, national and international. And having said that, I think it's turn for Mariona to do her presentation. I must just point out that we have some technical problems and we'll hear her perfectly, but we won't be able to see her, which is a pity, but I think sure that the topic of the talk will be fantastic. So Mariona, please go ahead.
Okay, Jordi, thank you for this nice introduction, and I'm really sorry that my camera is not working. I hope my pictures entertain you and you can follow my talk even if you don't see me. Okay, I know it's pretty difficult to follow seminars even online, and without an image or like seeing the speakers is even more difficult. But I promise you that you'll be entertained with my beautiful images. So as Jordi said, I've been working with the vascular compartment for a long time now, since actually I was a postdoc. And in fact, beyond that, when I was already a PhD student, which I was interested in understanding the physiology of the blood vessels. And I've learned a lot over the past 20 years. And I think I'd like to start my presentation by providing you key concepts that we've been learning over the past years. Okay, so so you all know that the vasculature it's composed by, so these blood vessels are tubes which are formed by a single monolayer of endothelial cells, and I'm sure you all know that there are this vascular network is composed by arteries, veins, and capillaries, and for very, very long time these vascular tubes were simply considered passive conduits for oxygen, nutrients, immune cells, and even to remove waste. But we've learned over these years that this is much more complex than that. And just think and imagine that it has to be more complex than that, because the endothelium is actually one of the few tissues that shows ubiquitous location, and it has the largest surface in the body. In fact, a human being of 80 kilograms has 3 trillion of endothelial cells, which weigh about 1 kilo. So we can consider that the endothelium is the systemic disseminating organ. Not only that, not only is ubiquitous, and it's like there's one extra concept about the endothelium. It's like, despite of being ubiquitous, it's different in each tissue. And this is an emerging concept in the vascular field, the angiodiversity of the endothelium, as you can nicely see in these beautiful pictures in which the blood vessels are depicted in different tissues. And part of this diversity, it's related to how this monolayer is organized. So we have the continuous, we have the fenestrated, and we have the discontinuous endothelium. And each of these adapt to the needs of the parenchyma. But there is even, okay, so I'm going to use my, see whether I can, okay. I was thinking about having a pointer, but I'm going to use my mouse here. It's not only different at the level of different tissues, the endothelium is also different within a tissue. And this is very nicely exemplified sorry, about the on the endothelium, for instance, of the bone marrow, in which we have different subtypes of vessels depending on the location of these vessels. So close to the metaphysis, we have the type H vessels, and in the diaphysis, we have the type L vessels, which have actually specialized for the different functions which are required in the hematopoietic differentiation process. And most importantly for me is the fact, what we're learning recently is that the endothelium is, as I said, is not this bystander tubes which like help distribute the nutrients and oxygen, but they in fact regulate organ function. And there are two modes in which the endothelium regulates the organ function beyond being just tubes. One is the anatomic characteristics of the endotheliums, which often serves as a homing for cells which live in close proximity to these endothelial cells and maintain them in this dormant state, as is the case for the hematopoietic stem cells. But also, the endothelium is a large producer of paracrine factors, what we nowadays call angiocrine factors. And these angiocrine factors regulate the physiology, the function of the tissue where these endothelial cells live. And these angiocrines are different depending on the tissue that this endothelium is located. So I think the first message of today is that over the past years, we've learned that blood vessels are not simply tubes, that they are highly heterogeneous entities. So each tissue has a subtype of endothelial or endothelial cells, and that they are emerging, this endothelium, these blood vessels are emerging as key and dynamic components that orchestrate the crosstalk between the circulation and organs. So in my lab, we're interested in understanding how these tubes, how this endothelium grows in development, and how we can translate the knowledge that we learn in development in these settings. And one of the key aspects we're interested in is understanding this growth at single cell resolution. We're learning nowadays how important it is to understand the single cell molecular profiles of these of cells in general in tissues. But we don't have to forget that we also have to understand how they look, how they organize, and how are they located in the tissues that we want to study. So this is a beautiful image of single label endothelial cells in which we can, we're very much interested in understanding how they function in a vascular growth context. And finally, we're also interested in understanding how this growth regulates the organ function. And the way we approach that, as I said, is in health and disease. So we use a variety of models that allow us to study development, the growth, the vascular growth and development, and also disease settings. And when it comes to disease, we approach it into flavors. In those diseases in which the vascular compartment plays an extrinsic role, so contributes to the development of the disease, and we also study those diseases in which the vasculature is the aberrant tissue, meaning the vascular growth process or pathological process is intrinsic. And the way we've been doing that in my lab for the past 10 years is focusing one pathway, the PI3 kinase pathway. This is a very simplistic way of showing you the pathway. I will go more into details later. But the reason we've been focusing on this pathway is because we've learned that the endothelium, and nowadays we know that it goes beyond the endothelium, the endothelium is exquisitely regulated by the PI3 kinase pathway. So we can use this pathway to understand how the endothelium works. So today I'll be discussing three concepts. First, I'll be showing you what we've learned, how the PI3 kinase regulate the growth of the vasculature during this angiogenesis process. I will also be showing you the importance of being selective. So there are multiple isoforms of PI3 kinases, but only some of them are important to form blood vessels and in a cell-specific manner. I will show you that too. And then I will also be discussing an emerging concept in the vascular field, which is the impact of oncogenic mutations as a cause of vascular-related disease. But before I'll show you real data, let me present you the PI3 kinases, which are somehow a bit complicated. So mammals have 8 isoforms of PI3 kinase, and these isoforms are subdivided in 3 classes based on, so basically based on the lipid that they use to phosphorylate, and also based on their structural characteristics. But they are all lipid kinases, and they all phosphorylate the 3 position of the inositol ring present in phospholipids in membranes. But depending on the substrate, as I said, we can classify them, right? So over the years, we've learned that these isoforms are much more complex than what we anticipated, because they not only use different substrates, but they also have different patterns of expression. They are activated in a different manner. They localize in different places in cells. They do have different physiological roles, and also mutations have been identified in several members of this family with different roles in disease. So my lab has been focused on the class 1, and in particular on the class 1A. But the class 1 is the subtype of PI3 kinases, which are acutely activated by extracellular cues. They are the only ones that produce the famous lipid PIP3, which activates AKT, and this lipid is actually negatively regulated by the phosphatase P10. So when you ever do a Western blot to assess phospho-AKT in your experimental settings, basically what you're analyzing is the class 1 PI3 kinases. And as I said, this is further classified in A and B, depending on which is the regulatory subunit that, so the dimers that form these complexes. So the 1A, it's composed by alpha, beta, and delta, and is an obligate heterodimer with P85, with the regulatory subunit P85. But let me dig in a bit more on these isoforms, and that's related to what I'll be discussing later. So we have 2 isoforms, the alpha and beta, so the catalytic isoforms. So in the PI3 kinase field, we tend to call, to use the name of the catalytic isoform to describe their functions. So alpha and beta are widely expressed, while delta is enriched in the hematopoietic system. What we know is that they are all acutely activated by extracellular receptors, but in a different manner. So alpha and delta are activated by tyrosine kinase receptors, while AP110 beta is a complex isoform which is activated by multiple inputs. We've learned over the years, mainly by using mouse models, that they have different roles in physiology. So alpha is important for metabolism and angiogenesis, beta is important for male fertility and platelet function, and delta, of course, it's important for immunity. They also play relevant roles in disease. And what we've learned over the past 5 years, I would say, is that the very same mutations that occur in cancer occur in monogenic diseases, with alpha causing what we call PROS, and I'll be discussing a bit more about PROS later, and mutations on gain of function, mutations on PI3 kinase delta, causing primary immunodeficiencies. So I'm going to be focusing on alpha because this is exactly how I started in the world of PI3 kinases. And this goes back to when I was a postdoc back in London, in which I was given the opportunity to, by characterizing a variety of mutants, to understand which was the isoform that is necessary for blood vessels to develop. And to cut a very long story short, what we found is that only the isoform alpha is essential for vascular development. And this story, which has had a lot of repercussion in the field, it made it into Nature for 2 reasons. First one, it's because we identified that this is an endothelial cell autonomous effect, so P110 alpha is required in endothelial cells to make blood vessels, and also because it's isoform is an isoform selectivity process. Only alpha regulates this process. And we found out this by targeting all the different isoforms individually. So when I moved back to Barcelona, my hometown, so the question I had in mind is that, okay, so I know alpha is important to make blood vessels, but I want to understand how alpha makes the job. And then what I'm going to be showing you now is that what alpha does is regulates a process that is called cell rearrangement. So cell rearrangement is a biological process in which collective cell migration happens. And as you can nicely see in this beautiful video, these are a monolayer of endothelial cells that you can nicely see in the green staining that these endothelial cells are attached to each other through cell-cell junctions. And endothelial cells use these cell junctions to propel the collective movement. And the way they do it is that by alternating weak like this and strong junctions. So they use the strong pattern to support into the substrate and the weak junction moment to propel the movement towards the gradients of growth factors. So what we found is that if alpha is inactivated in these endothelial cells, these junctions, as you can beautifully see them here, remain immature. They don't manage to make this pattern of weak and more stable junctions. So this is nicely quantified this. And then the impact of this, the fact that they remain immature is that they don't elongate the junctional contacts. They don't stabilize, they don't elongate the junctional contacts. And instead, they stay put. These endothelial cells don't move. They don't move, but they're still able to receive the signals to grow. And instead of moving, what they do is they keep elongating and elongating, as you can nicely see in this mouse retina of P110 alpha mutants. And eventually, instead of having a mature plexus composed of tubes, which have blood circulating on it, in red you can see the endothelial cell nuclei, you have tubes which are not perfused, there is no lumen, these are just protrusions of endothelial cells which don't move, don't rearrange. And through a massive phosphoproteomic screening, what we found is that PI3 kinase, P110 alpha in this case, regulates actomyosin contractility through the MYPT. MYPT, which is which dephosphorylate MLC2 and therefore negatively regulates actomyosin contractility. And the point I want to make here is that we tend to think that PI3 kinase, it's AKT, everything goes through AKT and mTOR. That's not true. There is life beyond AKT and mTOR. And if we want to understand how PI3 kinases regulate biological process, we have to remember that there are many other targets downstream of PI3 kinase, so we have to keep looking into them. So what I've shown you up to now is that too little PI3 kinase in endothelial cells is not good, because there is no proper formation of blood vessels. So there is a failure to develop the vasculature. What we later identify by using gain of function approaches is that too much is not good either, as these embryos here, in which we are constitutively activating PI3 kinase, they don't manage to develop a functional vasculature. So this highlights this concept I was trying to tell you before, that endothelial cells are extremely sensitive to PI3 kinase fluctuations. And that brings me to the second part of my talk, in which I'm sure most of you know that PI3 kinase, together with the p53, is one of the most frequently mutated pathways in cancer. So in fact, P110 alpha, which is this the protein that we've been discussing on its role on the vascular compartment, it's encoded by the PIK3CA gene. And amongst the PI3 kinase members, PIK3CA, so it's one of the most frequently mutated in cancer. And here you have a nice cartoon which illustrates this the gene with mutations that span the whole gene. But there's some hotspot locations located here in the helical domain of the P110 alpha, and also in the catalytic domain of the P110 alpha. So these mutations, I said, they're very common. So 30% of cancers contain mutations on PIK3CA, and they are somatic mutations. What we describe here in this Nature Communication paper is that these mutations are incompatible with its existence in the germline. And the reason is, I was telling you before, because these embryos don't manage to make proper blood vessels, and they fail to develop properly, right? What you might not know is that the very same mutations which occur on cancer are present in congenital disease, and they cause what is called overgrowth. And I'm gonna just show you a patient which has these mutations, which has acquired this mutation during development. And this patient, as you can obviously appreciate, it has an overgrowth problem. So this disease nowadays are known as PROS, or PIK3CA related overgrowth spectrum. And this is a very emerging field of a study in the field of PI3 kinase and beyond, not only PI3 kinase, because thanks to the next generation sequencing possibilities nowadays, or over the past 5 years, we've learned that several genes which are have been identified to be relevant in the context of cancer are also occurring in congenital syndromes. So PIK3CA is one of them, but this can be expanded to many others. So let me explain you a bit what these diseases are. So these are congenital, so these are mutations which are acquired during embryonic development and are not repaired, and they often occur in a mosaic fashion. So they're somatic, and only some cells within the body of these children contains the mutation. In the specific case of PROS, most of these cells, the cells that contain the mutation, are of mesoderm origin. So when this disease, when the presence of PIK3CA mutations as a cause of PROS was described about 6 years old, perhaps 7 years ago now, we were interested in, so it was a time in which it was already somehow anticipated that the PI3 kinase inhibitors in oncology were not doing so great. So that was the moment that we decided that perhaps, because we were interested on congenital, on development aspects, we thought that perhaps we could try to model this disease to provide a preclinical model for drug testing for this disease. So these are pediatric disease. Pediatric disease, I'm sure you, most of you know, being in a leukemia institute, are a very tough disease to propose treatments because of it's very hard to open a clinical trial with children. So you need to have the real proof that an inhibitor is going to work in order to jump first with a compassionate use of that drug. So we were, okay, let's try to model overgrowth. We were very optimistic at the time, but we decided to do that, but basically mimicking exactly what it happens to the human disease. So I told you this is a congenital syndrome of cells, of a mosaic scenario, and that the mutation occurs in cells of mesoderm origin. So what we did, it was, we used this TCRE-RT2 line here, that, in which the TCRE is a gene that is expressed in embryonic mesoderm, early embryonic mesoderm, and we cross this TCRE-RT2 line with the a mouse model that allows us to activate in the last exon of PIK3CA one of the hotspot mutations upon creativity. But before that, we tritiated the dose of tamoxifen to be used, to be given to the pregnant moms in a way to generate a mosaic situation. So we picked these two doses, the intermediate ones here, which give rise to different levels of mosaicism. So upon crossing with the PIK3CA, we gave tamoxifen to the pregnant moms. And the surprise was that that none of the mutants developed overgrowth. We were very disappointed, as you can imagine at the time. But what we found is that all of them developed vascular lesions, as you can see here. So the higher the degree of the mosaicism is, the more severe the lesions are. So as I said, we were very disappointed until we showed the pathology of one of these lesions to a very talented pathologist back in London. And he was actually the first one to spot that this looks exactly like a human disease that is called venous malformation. So venous malformation is also a congenital disease. It's rare, but it affects a large number. Within the rare is one of the most common. It's a very painful disease that may lead to bleeding and obstruction of large organs. And at the time that we started this study, there was no treatment other than sclerotherapy, which is not very efficient. And only 50% of the cases had been identified at that time with mutations on the TEC gene. TEC is a tyrosine kinase receptor, which is mainly present in endothelial cells, and it's an active PI3K kinase, right? So here you have different cases of people with venous malformations. So based on this data, we went back to the clinicians and we said, okay, is it possible that venous malformations are also caused by mutations on PIK3CA? So what we did was, through collaboration with clinicians, we managed to get a small cohort of patients. So here you see 13 patients, the pediatric patients, as you can see here from the age. And by doing targeted sequencing, very deep targeted sequencing, because this is a mosaic lesion, we could identify that 25% of the cases actually contain a pathogenic hotspot mutation on PIK3CA. So as you can also nicely hear is that these mutations are mutually exclusive with the green ones, which are those present on the TEC gene, the tyrosine kinase receptor I was telling you about. So this is a clear. So I like to tell this. So I like to say two things when I reach this point and I present this data. First is that negative data is never a negative data if you dig in enough. So we were very disappointed, but eventually this led to a total success. And the second point I want to make is that this is a story that comes from studying mice, and it ends up identifying or linking the biology with a human disease, right? And we feel very proud of it. So in the lab, we tend to use models which allow us to study in detail the behavior of these endothelial cells. So I just described you a disease that is caused by mutations in the endothelial cell compartment. So we want to understand, so our aim was to understand how these endothelials, which contain the mutation, behave. So we use different models, and one of the ones we use are the mouse retinas, which develop post-birth, allow flat mounting, and they provide us with these beautiful images that provide very deep insights about the behavior of endothelials. So here you have one of the petals of a mouse retina at postnatal day 6, in which we have labeled it through the use of 3ERT2 technology, single cells. So therefore, this allows us the single cell resolution that we need to understand how they behave. Not only that, so these technologies also allow us to study the degree of mosaicism. So how many cells do we need to get a lesion, for instance? And they are perfectly beautiful to study preclinical studies. So let me put you some examples. So here the aim is, can we generate a mosaic retina with isolated lesions induced by PIK3CA? The answer is yes. So here you have an experiment in which we have provided or given to the pup. So we inject 4-hydroxy directly to the pup, a high dose of tamoxifen. And in here you can see the massive hyperproliferative response that induces the expression of these oncogenic mutations on PIK3CA. While if we use a low dose of tamoxifen, actually we manage to get isolated lesions. And by having isolated lesions here, we can compare in one single preparation what happened to the lesion versus the normal endothelium, as it happened in humans. So if we do so, we can nicely see how these lesions only appear, if we use the mouse retina, they actually faithfully recapitulate to what happens to humans. So these lesions appear either in vein locations, as you can nicely see here, or in capillaries, but never in arteries. We can also assess how is the signaling in this lesion. So here you have a beautiful lesion, isolated lesion with high PI3 kinase. And we can also assess how the clonality, so the proliferative behavior, the clonality, and the composition of the lesions, whether they are multiclonal or coming from a single. And as I said, we can also use these retinas as a proof of concept for the use of PI3K. In this case, because it's caused by a mutation, but on PIK3CA. The idea was to test whether the PI3 kinase inhibition would work, but this can be extended or expanded to any new pharmacological approach that we wanna test. So in here, that was the proof of concept that we can rescue this massive hyperproliferative response with rapamycin. So I'm sure you know rapamycin is one of the safest drugs ever. So it has been tested for multiple disease and approaches, including oncology. It targets mTOR, and mTOR is a downstream effector of PI3K. So thanks that we provided the proof of concept that rapamycin works in preclinical models, and that the rapamycin is one of the safest drugs ever, we could jump directly into patients, pediatric patients in the clinic, through an ongoing collaboration that we have with Dr. Mora in Hospital Sant Joan de Barcelona. So as you can imagine, that was a super momentum in our career, in which we could see how the quality of life of these children has improved dramatically. So I told you that we were disappointed because we did not model overgrowth, but in fact, that was a full discovery in itself, because we identified that the very same mutations that cause overgrowth cause venous malformations. And what we've learned over the past 5 years is that venous malformation is actually a subtype of PROS. It could be included within this umbrella that we call PROS. And if I put together what I said just now about PIK3CA and the different subtypes of disease, so I started saying that these mutations cause cancer when they appear in adulthood, that they are incompatible with their presence in the germline, but during development, they can occur in a somatic fashion. And depending on when, so this is the important concept here, is depending when these mutations occur, these patients may end up having a dramatic PROS, as the kit I show you, or they simply have isolated lesions, such as the venous malformations I show you. So early mutation occurs early in PROS, in severe cases of PROS, while the mutation occurs late in isolated venous malformations. In fact, we know nowadays that vascular malformation is a common trait of PROS. So most of the patients that have a severe case of PROS have vascular alterations, and that we can subdivide PROS in 3 types: those which only have isolated lesions, those which have only one compartment affected, such as this kit here, compartment meaning only the vasculature is affected, but then it has different, this would be this one, but then it leads to a complex syndrome because there are different subtypes of endothelial cells which are affected, venous, lymphatic, capillaries. Or as these children here, that it has vascular alterations, was a part of a complex syndrome because not only endothelial cells are affected, but other cells of mesoderm origin are also affected. So since then, my lab has been expanding, and we've been growing, and we nowadays are interested in a variety of gain of function mutations which lead to these vascular alterations, either as affecting only one single compartment, as a part of a complex syndrome, somatic mutations which are acquired during development, but also those cases in which the presence of a mutation is compatible with the germline. And I just like to highlight that most or some of these mutations are the very same ones which are present in cancer. Also, we have expanded the way that we are approaching this disease. So through collaboration with different hospitals across the Barcelona network, we have developed a unique collection of patient-derived primary endothelial cells. So we get fresh biopsies. We've been isolating primary endothelial cells and non-endothelial cells, and we've managed to identify that the mutation is only present in the endothelial cell fraction compared to the non-endothelial cell fraction. So this, as you can imagine, this material is very it's unique, and it's very powerful for several reasons. One is because it will allow us to identify unknown mutations. So in some of the disease, there are still a large proportion of patients which are orphan. It's ideal to test targeted therapies and to study the biology that these mutations cause. And I'm gonna move to the last part of my talk, changing topics completely, and to show you more things that we have been doing in the lab and that can complement our understanding of how blood vessels form during development. So blood vessels are not, so I've been discussing a lot about the endothelial cells, which are the cells that form the monolayer of blood vessels, but blood vessels are much more than endothelial cells. They also have these, what we call mural cells, which are cells that are covering the blood vessels and regulate their function. So, depending on the type of blood vessel that we are talking about, these cells are called smooth muscle cells or pericytes. So smooth muscle cells will be covering arteries or arterioles, while pericytes are covering capillaries. So the field has experienced a boom over the past 10 years in the understanding of how endothelial cells regulate vessel growth. And this is in contrast with what we know about how pericytes, how mural cells regulate this process. And I told you they're equally important. If a blood vessel does not have mural cells or pericytes, it's useless because it cannot function properly. So we had this very simple question. It's like, what is the role of pericytes in angiogenesis? Do they regulate this process? So to study that, we use again our favorite model, the mouse retinas, but instead of using a the CRI-RT2 approach, but instead of using a line that labels endothelial cells, we use a line that labels pericytes, mural cells in general, because pericytes, you can see pericyte here, but we're also labeling smooth muscle cells. But if we make a quantification, this just by numbers, as simple as that, most of the cells that we're targeting are pericytes. So we decided to take advantage of this line in combination with the Ribotac approach. For those that are not familiarized with, the Ribotac is a tool that allows, by using the through the use of the CRI-LOX technology, to tag one of the proteins that forms the ribosomes. So in here we tag it with an HA tag, and then by immunoprecipitating, so using antibodies against HA, we can immunoprecipitate the ribosomes, and then all the mRNAs which are attached to these ribosomes. So this becomes an ideal tool to assess the translatome of a cell-specific, in a cell-specific manner in vivo. And this is exactly what we did using the mouse retinas, and we decided to confront two different time points. A very early time point in retina development, in which most of the vessels are, all the vessels are very mature, they're still going through a very early angiogenic phase that is called sprouting vessels, through a late time point, postnatal day 9, in which most of the vessels have already acquired what it's called the remodeling properties. So we perform RNA sequencing, and we, by of these samples, confronting these two time points. Of course, as expected, we identified lots of transcripts which were differently regulated both at P3 and at P9. And by performing gene ontology, we identify some aspects of these pericytes, which are very obvious. The first one is that by P3, in very early phases of angiogenesis, pericytes are highly proliferative. So there is a proliferative signature at P3 compared to P9. And we went back to the retina and we validated that indeed these pericytes are proliferating by imaging. And here you have P3, you have P9. You can nicely see how at P3 there are many more pericytes which are proliferating compared to P9. The beauty of the retina is that depending on the time point that we isolate, we can assess the behavior of these cells. And if we actually pick an intermediate time point, such as postnatal day 6, we can assess in one single preparation 2 types of behavior. If we focus at the front of the plexus, we can see that cells behave like at P3 as expected, but if we focus on the remodeling parts of this postnatal day 6, they actually behave like P9. So it becomes also a very useful tool. By focusing at P9, we identify 3 important signatures. The first one was which were upregulated. So the first one is related to the function. So there are many contractility genes. So in remodeling vessels, as the angiogenesis process advances, these pericytes acquire the more functional properties. And this is also linked to the expression of the canonical pericyte marker, indicating that they are acquiring their full differentiation properties. One thing that we did not expect is to identify an addition signature, as you can see here, at P9. So there were many integrins and extracellular matrix proteins, which the expression was upregulated by P9. So we were intrigued by this, and we decided to explore these phenotypes a bit further in detail. And we did that by using a reporter, the MTMG reporter, which nicely allows to label the shape of the cell. So we use, in order to identify single cells, we use very low doses of tamoxifen in a way that we could label only few. And we were very nicely surprised to actually see that these pericytes look very different at postnatal day 3 and at postnatal day 9. So at P3, they are poorly attached to the blood vessels and they have this spiky morphology, while by P9, they are elongated and properly wrapping or like the endothelium. So, so this is quantified here. And we also notice, I mean, we're a PI3 kinase lab, right? So we always tend to go to explore whether the PI3 kinase would be somehow involved in this process. And what we notice is that by P9, there is a strong upregulation of a FOXO signature. So FOXO is a negative regulator of PI3 kinase. So if a FOXO signature it's up at P9, it means that PI3 kinase is low. So we decided to validate this, and to do that, we took advantage of this line, the Hey1GFP, that it's a line that labels immature pericytes. Here you can see the triple staining. In blue, you see the endothelium. In red, you see the canonical marker of pericytes. It's a canonical marker, NG2. In green, you see the immature pericytes. And in white, you see we use phospho-S6 as a readout of PI3 kinase activity. So this is a downstream effect of N2. And what you can nicely see in this image, and we also did some quantification, but what you nicely see is that those pericytes which are located in the sprouting front, which are immature, so-called immature pericytes, have a high PI3 kinase signaling compared to those located in remodeling vessels, as the molecular signature had actually identified with a low PI3 kinase in the remodeling or mature vessels. So, sorry, what's this? So, if I summarize what I've been showing until now, is that basically those pericytes which are located in sprouting vessels, we decided to call them immature pericytes. So they are pericytes which are proliferative, they have this spiky shape, and they are exhibit high PI3 kinase activation. Instead, those pericytes which are located in remodeling vessels we decide to call them mature pericytes. They are quiescent, so they rarely proliferate. They have stopped proliferating. They're highly elongated. They properly wrap the blood vessels, and they are low on PI3 kinase. So the obvious question is, what's the impact of altering the PI3 kinase pathway in pericytes? So if we take pericytes in culture, they all express the class 1 PI3 kinases that I've been introducing you before. And if we use selective inhibitors for each of the isoforms, you can nicely see here that only alpha and beta impact on the activation of phospho-AKT as used as a readout of PI3 kinase. So in the lab, we have available tools which allow us to inactivate alpha, beta, and P10. And that's what decided to do, to study if we inactivate one or the other, if we actually alter the behavior of endothelium, of pericytes. So to cut a long story short, I showed you before that alpha is essential for endothelial cells to expand and form blood vessels. Instead, we didn't find any role for P110 alpha in pericytes during angiogenesis. But the nice surprise was that if we inactivate P110 beta, we have a strong phenotype. And the same happens if we inactivate P10, which would mean that we would be chronically activating PI3 kinase. So what I'm going to be showing now is data on inactivation of P110 beta, deletion of P110 beta, which means inactivation of the PI3 kinase pathway, and I'm going to be focusing at postnatal day 6. a time point in which I expect, based on what I've learned in wild-type retinas, that pericytes are still immature in some of the areas. And then later, I'll be focusing on P9, a time point in which pericytes are largely mature, are expected to be largely mature. And I'll be deleting P10 at this time. And this is actually what happens. So if we inactivate P110 beta or inactivate PI3 kinase, instead of having immature pericytes, these pericytes are behaving like mature pericytes. They are quiescent and they have this elongated shape. Instead, if I activate PI3 kinase, these pericytes rarely acquire the mature properties, even at those time points in which they are supposed to be mature. And you can nicely see here by their proliferative behavior, but also by their shape. So these pericytes sustain or keep this stellate shape, and they rarely acquire this nice elongated shape required to properly attach surrounded. We verify that by assessing the molecular profile of these pericytes in vitro, again by using the ribotype. So if we inactivate PI3 kinase, there is an enrichment of the postnatal day signature, even if we are at postnatal day 6. And if we activate PI3 kinase, there is an enrichment of the P3 signature, even if we are at postnatal day 9. So inactivation of PI3 kinase sustains the or transform immature pericytes into mature pericytes. But if we activate chronically PI3 kinase, these pericytes remain in an immature state. So the obvious question, and this is how we actually started the whole project, is that do these two phenotypes impact on endothelial cells, are relevant for angiogenesis? And the answer is yes. So mature pericytes, which we obtain if we inactivate PI3 kinase, render endothelial cell quiescence, as you can nicely see here by the density, but also by the proliferative behavior of these endothelial cells. And the opposite is also true. If we sustain the immature state of pericytes, they actually stimulate, continue promoting the endothelial cell proliferation, as nicely seen by these images. And this is translated into a more dense vascular plexus. So overall, what I just nicely show you is that the pericytes are important for angiogenesis. They are required. They actively participate in this process. They are that the state of the pericyte instructs the behavior of endothelial cells, that the PI3 kinase pathway is actually key to modulate the mature state of pericytes, and that we believe that the pericyte maturation could be used to manipulate vessel growth in pathological conditions. So this is my last slide, and which nicely summarizes actually the take-home messages of my talk. So the first is that I show you by using the PI3 kinase pathway, a pathway that that regulates blood vessel behavior profoundly. That I show you that PI3 kinase regulates endothelial cell rearrangement, so this intercalation of the endothelium during the growth of the vasculature. It does also regulate the behavior of the supporting cells, the pericytes, because it sustains the maturation state. And what I think is very important from my talk, and I hope you take this with you, is that this is isoform selective. And the evolution system has managed to identify different isoforms which cooperate together for the regulation of the formation of an adequate vascular plexus. And finally, that of course P110 alpha is so important. It's not surprising that gain of function mutations are on the gene that encodes for P110 alpha are one of the main causes of venous malformations and in general of vascular malformations. So there is still a lot to learn, and I hope we can gain insight with in the future to understand better how this process works. So I have to finish by thank you those which have done the job. So this is a combination of team effort of current members and past members of my lab. So thank you. They are the real start. Thanks also to all the agencies which are supporting my work, past and present, and the collaborations. Without them, I will not be here. And before I finish, just remember, if you like vascular malformations, we're holding a specific ONBO workshop on this field. It's going to be super cool, so don't miss it. And I'll be happy to take questions.
So thank you very much, Mariona, for this very nice presentation. I think it's really inspiring. As you know,
I hope everybody's still here as I
think so. So we have some questions in the chat. So perhaps just I can read some of them. One of them is there an interest of PI3K related inhibitors combined with standard anti-angiogenic therapies when there is a mutation in the pathway?
It's an interesting question. So it could be, why not? This has not been tested. I've never tested, like for instance, an anti-VEGF inhibitor with a P110 alpha inhibitor. It's true that they block the growth of the vasculature in a different manner. So this could be tested. Although the angiogenesis therapy as such, at least in cancer, has been developing in a way that it's trying to identify through which ways the endothelium can be converted into a better endothelium and not destroying the endothelium as it was suggested in the original days of the angiogenesis therapy. So this should require a deeper, at least brainstorming to see which would be the best way to approach this combination. But I've never done it.
Okay, thank you. We have another question. Since PI3K and mTOR are directly associated to metabolism, can diet modulate vascular malformation?
Wow. This is an interesting, definitely. So the first answer that comes to my mind is yes, for sure. We do know that diet actually interferes with the endothelial cell behavior. And we do know that this, the way that interferes with PI3 with the behavior is through PI3 kinase. Whether this is going to influence the growth of the vasculature when a mutation is acquired, I don't know. Be interesting to test, but I would say yes.
Okay. So we have another question with some congrats. Great talk and exciting data, Mariona. Congrats. What is the basis for the isoform specificity of PI3K alpha? Expression pattern or difference in the protein or something else? Or asking the other way around, can other PI3K isoforms rescue PI3K alpha deficiency if put under control of a PI3K alpha promoter? I think it's an interesting question also.
So the reason of isoform selectivity, it's a combination of several things. First is alpha is expressed, but so it is in the endothelial cells is expressed, but so are the other isoforms. So amongst the three I was discussing, alpha, beta, and delta, alpha is the most active and is the preferentially activated by RTK receptors in endothelial cells. And RTK receptors are extremely important for the growth of the vasculature. So that would be the easy answer. Whether other isoforms can play a role when alpha is gone, my guess would be yes, but this has not been tested because PI3 kinases are very tricky proteins to work with, and one has to use very specific approaches in order to avoid the interference and the overlap of the different members of the pathway. So these all my experiments have always been done in conditions, in inactivating conditions and not knockout conditions. So I have not done this experiment, but I think if we would try to overexpress, for instance, massively P110 beta in endothelial cells, some of the functions may be slightly recovered. Not all of them, but some, yes. Yeah.
Okay, thank you very much, Mariona. I think we have time for one or more questions. We have now one in the chat. It's excellent, Mariona. PI3K alpha is involved in the junction stabilization in endothelial cells. Do you know if this is also true in cancer cells and metastasis?
Whether it plays a role in epithelial cells during collective cell migration, I don't know. It's worth exploring, but I don't know. I would say some of the functions that we have actually identified in the endothelial cell compartment have later been described in epithelial cells. So yeah, could be, definitely, but I don't know.
May I ask you a question about my topic, my field, which is leukemia, acute leukemia? Do you have any information in acute leukemia about if there is an activation of the vascular component of this signaling pathway? And do you know, or do you think if this can be a target?
So in hematopoietic cells, alpha is not important. It's delta, the isoform that it's relevant for any. So it's the isoform that regulates PI3 kinase signaling, and it's deregulated in the different spectrum of hematopoietic malignancies. And in fact, P110 delta inhibitors are in the clinic to treat some of these conditions. Having said that, endothelial cells are extremely important for sustaining the hematopoietic stem cells. Like they're homing, they are the wardens of these cells. And it's believed that they do play a role in the deregulation of these hematopoietic stem cells. So yeah, I'm really excited about being here in this institute, because I think my next step is to explore how endothelial cells, from an endothelial cell perspective, how they contribute to the dysregulation of these cells in these malignancies. But that's for my next talk, I promise.
Okay, so thank you very much. We will be expecting that talk, sure.
I'm sure.
For me, it has been very interesting. So thank you very much, Mariona. I think we don't have time, no more questions, but we will have time to think about all you have explained us, have told us. So thank you very much, Mariona, and to all the attendees for your participation.
Thank you, Jordan.